All right, so let's continue. Now that we're inside the mitochondria matrix, we've crossed over the two lipid bilayer membranes, and we're in the inside. So acetyl coenzyme A, our 2-carbon compound with a helper, crosses the membrane and gets inside, and coenzyme A leaves to repeat the process. That gives us a 2-carbon compound which joins a C4-carbon compound to make citric acid. So this is the beginning of the Krebs cycle. Again, the people who figured this out got the Nobel Prize. But we're not going to focus on all of the 10 steps in this process. We're just going to focus on the products. And this could be easily summarized by the 3-2-1-1 system. So we're going to start off with 3 NADHs being produced. Of course, NAD comes in, pulls off the hydrogen. The next thing that we're going to release is the 2 carbon dioxides, which of course we breathe off. They go into our red blood cells, to our lungs. And finally, we make 1 ATP and 1 FADH. So this is the final disembowelment or deconstruction of the glucose molecule. Nothing is left but to make the ATP. So let's summarize what we've made from the Krebs cycle in the matrix. So the Krebs cycle actually turns twice per glucose molecule, and in reality all of these reactions are happening simultaneously, but given that we like to think linearly, we've done it in a nice circular pattern. But we've summarized 3-2-1-1. 3-2-1-1 gives us 6 NADHs, 4 carbon dioxides, 2 ATPs, 2 FADH2s. So that's the final deconstruction of our glucose molecule. And let's add in our summary from glycolysis. Remember what we did on the previous video. We have 4 NADHs, 2 carbon dioxides, and we net 2 ATP. Now we made 4, but we only have 2. We needed to use 2 to start the process. So now we're ready for the electron transport chain. The only thing that we care about now, because the ATP is being used, the carbon dioxide leaves to the red blood cells. So the only 2 things that are left to be recycled or reused are the NADH and the FADH2.